Thank you to all the speakers. Even putting together a 10-minute talk is a lot of work and takes a lot of thought. So please make sure to give a pat on the back to the folks who spoke at the lightning rounds. And now I'd like to invite all of the panelists to please come up to the front. You should feel comfortable to sit or stand. We'll have a couple of mics in the front and a couple of mics roving through the audience. And we could use a couple of volunteers to help with roving mics. Would a couple of people volunteer? Excellent. Thank you. While we're working out how to use the mic, if you have a booming voice and you'd like to stand up and ask an initial question, go for it.
This question is for Janelle and Mia. Why are you seeding into basins? And then would you consider doing more broadcast seeding?
We would consider that, and I think the reason we're doing basins is so that, at least for this particular project, it's so that we can more accurately track the results. So we put 4 seeds in each basin, and we're looking at percent germination, and we're also looking at survivorship and health and vigor over time. So it's just an easier way to contain a little site for statistical analysis, and also because we have irrigation set up to each basin, it's easier to track it. When we were first designing this, we did think about maybe just doing some sort of a bed type where you just throw out a bunch of seed, and that would be a truly more natural random situation, but it was harder to evaluate the results, we thought. So that's why we haven't yet gone to that type of a design.
Yep. For us, it was because we had the irrigation, the infrastructure, everything already set up to these basins. There are a fair amount of areas, grasslands, where we hydroseeded a bunch of forbs and grasses, but for these woody plantings, we've been talking about changing our design in the future and maybe kind of doing rows in a way, but our whole irrigation, everything, tracking germination percents, survival, all of that is based around this certain design, but it's not optimal for seeding at all. And then also they have, they're trying to put tubex around these, and I'm like, you can't seed into a tubex that's this small and expect something good to happen. So we've been putting those on later once the plants are getting a little bit bigger, but it most certainly is not the best fit. And I don't think that if we redesign a new restoration project, we would do it in the same way.
Yes. All the presentations had really good findings for specific plant species, and then which specific Phytophthora species would affect them, and how that interaction went down in the field. Is there a website where people are dumping all these findings if we wanted to kind of sniff around and research on them?
Okay, so there's a few places to go for information. Mia mentioned that Matteo Garbalato and I and another author put together that review paper. I guess the review paper, it's in review, it's being reviewed, so it will be ready soon. In the meantime, I recommend Jennifer Park, that's here from Oregon State University, has the Forest Phytoptheras of the World website, and I think that one's very good. Probably the best place to go for now. But it is true that Jennifer, what's your website? Is she here?
Do you want?
Of the world, and it will come up. Forest Phytophthora of the world. You have to spell Phytophthora, but this group knows how to do that.
Yeah, but also I'm just noting that a to-do would be to make sure that that is on a website as well. the information that we've collected here in the Bay Area that's represented by the work from this group.
Thank you.
Oh, yeah, we do have a website for this group, www.calphytos.org.
You know, the earlier talks convinced me this kind of like a Mediterranean zone issue. Yet we talked about California native plants. I know that in agriculture there are some papers that have suggested broader use of other Mediterranean zone plants because of a variety of reasons and so forth, to use them for pollination, you know, to attract pollinators and so on. So is the name kind of restrictive? Or is our focus kind of restrictive because we're talking about California native plants? Should we have a more broad scope in terms of Mediterranean and drought-resistant plant use?
I'll just take a stab at that. Phytophthora are essentially an issue well beyond just Mediterranean species. I mean, agricultural species, horticultural species, you name it, there's Phytophthora going after everything. So our focus here is this is a restoration group in particular, and so that's where the focus has been on, where people are planting native plants for restoration. That's focus. But we have to, I don't mention it in this particular talk, but whether we're talking about native plants or Mediterranean zone plants grown in commercial nurseries, there's no firewall between native plant nurseries or there hasn't been, between native plant nurseries and commercial nurseries. So if native plants are grown in commercial nurseries, that's an issue no matter what, and if vice versa. So we just need to keep in mind that's why we've got these BMPs to set aside a different segment where we can produce clean plants.
Okay, so more questions? Okay, I did have another comment. So I think phytophthora occur, as Ted said, on all kinds of plants all around the world. And really, in Mediterranean-type climates, they are showing up more in the wildlands. And with any disease of a plant, it's a fight. The plant is trying to stay alive, and the pathogen is trying to do its thing. So the Mediterranean climates with their hot summers are a big stress on the plant. And I think that is really why, the phytophthora are out there in the forests everywhere. But when they kill the plants is the areas that have extreme drought stress.
There's a mic over there.
I had a question for Suzanne and Ted. It's interesting that you mentioned that grasses and other monocots are now showing up as hosts for Phytophthora. And I was wondering if you could talk a little bit more about that. What species are you finding on those monocots, and what are the plant species as well?
Well, I guess I'll start. The most, and actually Tyler probably mentioned this, we see a certain amount in Juncus seems to be a pretty hot for Phytophthora. We've picked up a number of species there, including one of Tyler's novel species that's shown up in that. Some of the grasses, we've actually found Elemus triticioides in a prop bed that had Phytophthora gregata associated with it. So there are clade 6 species in some cases, other cases, other things. So I'd have to look at our list. It's not a huge chunk, but the point was before we were kind of thinking, monocots, we don't care about them. And now they're on our radar as certainly, at least in certain families of monocots, it's not that uncommon. For that matter, Phytophthora erythroseptica we picked up in really just sort of a grassland area, and that's previously been described on rice as a rice pathogen. So, there's a number of things out there that do go to just straight grasses.
Yeah, we didn't. Most of our detections from grasses, a lot have been from TED samples and also samples that have been flow through testing. We don't pick up as many direct isolations from roots. I think one of them was a yucca relative, aerial yucca. That one we've picked up a couple directly from the roots, but a lot of them are associated with the roots and the rhizosphere. So I'm wondering if they're kind of maybe carriers too for Phytophthora too, or maybe they don't get infected as well as them, but they're able to support them in their root system. Just a thought.
One follow-up on that. If Giles Hardy and his people down in Australia have found cinnamomi will cause asymptomatic infections on some of their native grasses there. So in some cases, they're infecting these things, they're holding over, but not causing symptoms in those plants. So they may be reservoir hosts. For all we know, Phytophthora are really just bad pathogens, weak pathogens of grasses. But when they get on dicots, they're much more aggressive. I mean, we really don't know the original origin of most of these species and what their ecology is, and it may be that the fact there's such hot pathogens on such a wide variety of dicots is because that's not really where they started out. Maybe they really are adapted in sync with some other species.
I just want to add to what Ted said about the pathogen they found on rice, that there were rice wattles nearby. And we import straw, rice wattles, all kinds of erosion things, gravel, mulch, compost. So we're really looking at like everything that we import. It's not just container plants, and realizing that all of those things quite possibly carry the risk of introducing Phytophthora into our watersheds.
I actually have two questions that are completely unrelated. The first one for Diana. The heat treatment that you're doing in your buckets, are you treating soil that you've used before, or are you treating new media components as well? And
Yeah, it's new. We're not reusing any media, so
Okay.
it's what we get delivered to us and dropped off. We will.
okay,
heat
and
treat.
you're suspicious. Well, you're just careful enough that you don't know what the chain of locations or how it's been stored, that you just don't want to take any chances, and that's why you're pasteurizing or
So
the new
it's
media.
a potential vector, and
Uh-huh.
it is part of the BMPs from the Phytophthora working group. So
Okay.
certain clients will request that we add that BMP into their material. So
Got it.
our goal is someday everything at the nursery, but
Uh-huh.
we're not quite there yet. But for
Okay.
these particular projects.
Okay, and my second question, which, like I said, is totally unrelated, and it may be a can of worms, but thinking about introduction into natural area, forest areas, has anybody looked at sites where people have illicitly planted cannabis? And is that a source of entry into some of our natural areas? Because we can do all we want, trying to get BMPs for, legal activities, but surely, these plants also carry root pathogens, and they're being put in areas that we care a lot about, So does anybody know anything about that? Is that on the radar anywhere?
Well, actually, and there's a lot of severe impacts in areas where cannabis growers are going illegally in and doing sorts of things. And certainly, they're not terribly fastidious about what they're doing in almost any way, shape, or form. So, yeah, the possible introductions is that's certainly a possible route. I don't think so, unless and there's some maybe we'll get some big funding for that somewhere. Yeah. That's every watershed has got sites that they could be sampled.
I'd like to ask the panel, if you have a restoration site that's at a high risk for Phytophthora spread and you're trying to monitor and contain it, how often do you sample? What's your sample frequency and methodology?
So you're talking about a new site where you're going in. It's still tough doing baseline sampling. That's actually one of the few things we've been doing through this project with the Santa Clara Valley Habitat Agency, a Fish and Wildlife funded project. It's just going to sites and looking for what we can detect out there in sites that haven't had anything but just general impacts of people and grazing and whatever. And it's very difficult to find anything in those sites, partly because it's not out there. So in some respects, there's setting some baseline by essentially sampling areas, looking at the highest risk subsites within those sites is a reasonable way to go about it. And we're also sort of working on some methodology of sort of defining where the risk pathways are. And we certainly know roads and trails and things like that where contamination can be brought in is a potential risk pathway. But unless you've got a contaminated source nearby, that pathway you have to have all these elements. You have to have a source, you have to have a pathway, you have to have a sink, basically. And if all those three things don't come together, you don't end up with disease. So up to a point, I don't think there's a routine sort of method for doing this. I think you evaluate your site, what your risk pathways are, and from that you set up a program to say, okay, this is what we should do to make sure the site's staying clean.
Just one.
That for contamination to be brought in is a potential risk pathway. But unless you've got a contaminated source nearby, that pathway, you have to have all these elements. You have to have a source, you have to have a pathway, you have to have a sink, basically. And if all those three things don't come together, you don't end up with disease. So up to a point, I don't think there's a routine sort of method for doing this. I think you evaluate your site, what your risk pathways are, and from that you set up a program to say, okay, this is what we should do to make sure the site's staying clean.
Just one. We're going to talk about the working group work plan for the future and the point that you brought up that we need protocols and recommendations for monitoring and testing. That's one of the highest items on the to-do list. So hopefully from 2 years from now, we'll be further along.
Ted, this is a follow-up to what you were just talking about. I'm just wondering, in native habitats, are there ways that you would define certain habitats as higher risk versus lower risk in the field for places you're considering planting into?
Yeah, and we're right now working on a whole document for a GGNRA on that. And I mean, what we do know is that a lot of woody plants have a higher risk of infection than grasses. I mean, we can pick things up occasionally in grasses, but if you go out there and sample, you can sample a lot of grasslands and never pick up Phytophthoras compared to going to the adjacent woody vegetation showing symptoms and pick them up there. And we can sample outward from woody vegetation that's infested out into grasslands, and detections drop off to nothing. So we do know that the highest populations and the greatest impacts are going to be seen in these woody plant species. We do know some species like manzanitas and ceanothus and various other species tend to be really susceptible. So, and the other issue that really comes into is root connectivity and routes of movement. And so if you have widely scattered plants, one gets infected and that there's no root contact for 20 meters around it, chances of that spreading in that system is very low. Whereas if you have a contiguous chaparral or something where there's root-to-root contact continuously, root-to-root movement of the pathogen occurs quite readily and is mostly unstoppable. So there's a number of different factors, and connectivity is a big chunk of that differentiates a higher risk site from a lower risk site.
This is regarding soil in the nursery. Have you tested the efficacy of the heat sterilization treatments? And is the method that you're using, I guess I'm wondering, why don't you use soilless, sterile growing medium?
The growing media we use is soilless. It is not currently possible to purchase sterile media that's been treated. We've been trying to get some local distributor to be willing to take on that task, but we're not a big enough lobby yet to convince them that it's worth their investment in the equipment. So that's, in regards to that question, it is soilless, and it still is a potential vector in there, even though we've researched the origin of the different components of our mix also. It still goes to a yard, sits at the yard, gets in a truck, travels around, so it's a potential vector. So it's a best management practice to prevent that potential vector. In terms of testing the efficacy, my understanding, and I will let the pathologist take this over, is that testing just a batch of soil is really difficult to get enough isolate in without having a plant infected or something like that to have the organism amplified. So we have not done any testing of our soil pre and post for Phytophthora.
You want to? Yeah.
I've been getting a lot of questions recently about the difference between sterile media and pasteurized media, and I thought it would be good for someone. I'm going to hand it to you. Want it, Suzanne? No? Ted? To maybe explain the difference between pasteurized and sterile media and which would be preferable.
Okay, I'll take this, but I prefer the term just heat treated, and really the bottom line is, and kind of get back to your original question, there's a lot of research indicating what the thermal death points of various organisms are. So once you reach those thermal death points, as long as you can document you've reached them for the sufficient period of time, you don't really have to test the media. I mean, when something goes into an autoclave, you don't actually test the material out of the autoclave. What you test is the strip that shows the autoclave got hot enough to actually sterilize the material. And now, in terms of sterilization, that talks, if you're sterilizing something, you're killing all organisms. You have to go to high temperatures. Basically, you're going over the boiling point is what it comes down to, or under pressure, et cetera. For soil, we don't need to do that. Well, the pathogens basically can be killed at heat treatment of about 50 to 60 centigrade at the 150, 160 Fahrenheit, if I haven't got my numbers jiggled around too much. The bottom line is there's a death point in there which takes out the water molds, which actually are sensitive to lower temperatures, around 125 Fahrenheit. But then most pathogenic fungi start also going out at these higher temperatures. So we're not killing everything in the soil. A lot of bacteria survive, actinomycete spores, things like that. And that's not a problem. And so we're not dealing with actual sterile media. What we're dealing with is heat-treated media, heat-treated to a given standard known to kill plant pathogens. And that's the bottom line on this.
Is there a benefit to having those other organisms in there that don't sterilize?
Yeah, that's one of the things that people have kind of not wanted to do heat treatment because they think their soil's got all kinds of magic stuff in it. And soil does have sort of magic stuff in it in a way. However, potting media, isn't really soil, right? It's peat moss and forest product and vermiculite or sand or whatever. So what's the magic mix in that to begin with? So in some ways, I'm not really too concerned about if we kill everything in that media in a way because there's nothing really super special in it to begin with or anything that's really ecologically significant relative to where you're going to plant it. It's not the mix of organisms that exist in the field where you're going. So in that respect, people start material in perlite, which is a perfectly sterile material that's produced at very high temperatures. That's not an issue. People worry about mycorrhizal inoculation, and the fact is a lot of the VA mycorrhizae are very readily dispersed in air, so it's hard to actually keep them out of your plants. So in fact, introducing them is kind of a little bit superfluous anyway. So the bottom line is the heat treatment, there's no point in going higher than you need to just because it's energy, and energy is cost. So that's why we top it out at that. Also at high temperatures, you can have chemical reactions that cause problems with toxicity.
I just want to follow up. You gave an example of Phytophthora and pickleweed and whether you've seen it in other saline or brackish areas, and if you can just talk a little bit more about that.
Yeah, we don't have a lot of information on that. We have seen Humicula, the same species, in some willows that were declining and oaks. We have a sort of a saline area near Vacaville we sampled, which is got high salinity, and we picked up a couple of odd species associated with some plants material there. There actually had been a nursery in that same area, which is probably a source of all kinds of stuff. But we don't really know that much about that. We do know that I did work on salt tolerance of Phytophthora species for my doctoral thesis, so we actually know a lot about the salt tolerance of various species. It can actually vary from isolates. You can have isolates of Phytophthora nicotianae, for instance, that are relatively sensitive to salt, and other ones that are very highly tolerant. So the bottom line is, we kind of hope that when all these Phytophthoras got flushed into the bay, the salinity would go up, and most of them would be essentially unable to function, and that's probably the case. But over time, there are ones that have become adapted to or that are pre-adapted to these more saline environments and they are problematic if they're in sites where you've got susceptible vegetation.
I have two questions. One is, how many of these species of Phytophthora are actually naturally occurring in California, and how many are introduced from outside of California? And then my other question is, when you're using soil sterilization methods at restoration sites to get rid of infestations, and then also using the heat injection auger, is there any before and after studies done on how that's affecting other soil microorganisms and biota?
Okay, so for the first question, Tyler, can you come up? So this is Tyler Beret from UC Davis, the Rizzo lab that's been mentioned many times, and Tyler will be speaking after the break. But I think he's really the best person in the room to answer the question about the native Phytophthora.
It's one of the most fundamentally difficult questions that we're all dealing with, and the answer is it's for reasons that I'll go into in my talk. It's really hard to say even one single species is native or exotic at this point. In fact, we have a bigger list of species we know to be exotic to California than really ones that we know ones that are resident, but that doesn't necessarily mean that they're native. So certainly I could give you some examples, but it's a really fundamental problem that affects everything from the scientists to everybody, working on these problems. And I'm working on it. We're all working on it. But it's hopefully I make the case, it's a really difficult question.
Right, but I'll add on to that. From sampling that we've done, I mean, if there are native Phytoptheras, and they're not all just naturalized from being brought in, they're going to be from wet habitat sites, because that's really where Phytophtheras find their original niche. They are capable of functioning in drier sites, but we go out and sample wildlands at large, we don't find Phytophtheras just sitting everywhere. So there's not grand populations of native Phytophtheras out there. What we're really worried about is the ones that we're bringing in with our stock. And by now we've all forgotten the other half of the question.
The second part was the soil microorganisms using soil sterilization and the heat injection auger. Is there any before and after studies being done to see how that's affecting other microorganisms or soil biota?
Right, and one of the issues here is what we've got are essentially general habitats that were largely clean, where we put these spot infestations in. So the whole point of heat auger types or spot solarization is to try to eradicate the pathogens from these small areas. We're talking about a meter square, or for the augers maybe even smaller. So really, in some respects, we don't really care if we kill every single thing in that little bit of soil, because it'll become naturally colonized by the surrounding microorganisms anyway after the fact. But what we don't want is to allow that Phytophthora to persist in there. So we're treating so small a fraction of the total area that it's in biological terms, it's really not significant in that respect. That particular site, that was a holding nursery site. Solarization, there's lots of studies on it. The bottom line is it doesn't get hot enough, deep enough to really sterilize anything. The upper surface, you might kill a number of things, but it's more like pasteurization or heat treatment. You're not going to affect most bacteria, actinomycetes, a lot of soil fungi, and those coarse things. There's a whole body of solarization literature, and we're lucky to, if we do it right, to get the pathogens we're after.
Jennifer Park. Jennifer, where are you? Did you want to add anything, comment about the solarization and the sterilization? And
I'll talk about it. Yes. So I'm with MidPan, and a lot of our restoration sites may have endemic Phytophthora, not only because perhaps they're native, but also by their very nature, our restoration sites are disturbed. We don't do a lot of mitigation. We do replanting at previously disturbed sites, or we do replanting for a parking lot, which we've put at a site that is already disturbed because someone else put, a home on it 100 years ago and brought in, nursery trees from the East Coast perhaps. So we have, I think, in many of our sites, endemic disease, soil-borne pathogens. And I know this is a little off topic since we're really focusing on nursery practices, but has there been thought into developing a set of BMPs to avoid or minimize the conditions, the environmental conditions within these sites that may have endemic Phytophthora to prevent their spread or their activation on restoration plantings?
Right. And so I wouldn't use the term endemic. If you have an introduction there, whether it's recent or whether it's past, 100 years ago, and there aren't a lot of 100-year-old infestations. There are maybe 50-year-old infestations and 80-year-old ones here and there, and a lot within the last, earlier than that. Also, a disturbed site doesn't necessarily mean Phytophthora infested, as I kind of what we're getting to with our sampling and for instance, that Anderson Dam site, there's a lot of disturbance along trails and roads on areas there that there's no Phytophthora detectable in those areas. So if you don't have a source of introduction, the disturbance itself doesn't obviously create the Phytophthora. You have to have a source of enough intensity that it can actually contaminate the site. If you do have a contaminated site, then we're into what we call contaminated site protocol. So if you know that you have an infestation from some source, you really are looking at minimizing spread from that source. And there are BMPs that have been developed, and we're still working to increase the body of ways to manage those. But basically, yeah, your main priorities there are just not spreading it out of that site and not doing anything to increase the likelihood of spread. Treatment within sites is another option for very sensitive sites. We're doing this on Ione Manzanita, where we have essentially the habitat's being totally destroyed. We're using phosphite application at the borders to see if we can maintain a line to keep the infestation from going forward. So there is information in that line, but I think you can't make the assumption that every site is infested just because it's a disturbed site, or you're going to end up putting more effort in some cases than you really need to. So some categorization of the risk and some testing is probably in order, at least in some sites, before you go full bore into high level BMPs, unless you just really know that there's a high risk, like a former Christmas tree farm or something like that, where there's, yeah, and I know you guys have those. So those are sites you can assume probably do have infestation.
I think really briefly, I think that's a really big one. These are Mediterranean plants. Perhaps they don't expect to encounter Phytophthora during the summer. So whether you irrigate and how you irrigate is a really big thing about either if they're already there or if you planted them on one, having them spread to other outplantings. I think that's one thing we're finding out is, Mediterranean plants perhaps they don't have to worry about Phytophthora during the summer because everything is totally dry. They go into a dormancy, as does their native endemic Phytophthora. But we create a situation either in the nursery or out when we're irrigating where they've got pathogens at a time that they're not expecting them, and that's a potential problem.
So kind of building off Lisa's question, as a soil scientist, I'm interested in hearing more. Have you looked at general soil health? And are there bacteria or other fungi that can actually compete with this particular pathogen and build some resistance in the plants that are coming out of nurseries?
Yeah, there's plenty of literature, especially from Australia, about suppressive soils where, and actually even just on two sides of the slope, a wetter, more organic soil in that case, the south-facing slope will end up being suppressive, and though Phytophthora is there, you won't see severe disease, whereas on what would be the north-facing, our equivalent of a south-facing slope, it's much more intense. So you can have highly diverse microorganism populations that will suppress at least some Phytophthora. And the question is, we start getting 50 Phytophthora species out there, whether they're equally antagonistic against all of them. But you also have sites like Untitled Chapter, serpentine soils, where you're not going to modify those soil types. They're going to be innately poor and not have a lot of microorganisms in them, not going to have a lot of organic matter. And if you alter the habitat, you no longer have the habitat that you wanted. So what we have to, in many cases, is work with what is out there. In agriculture, yeah, you can go in there and modify. You can add organic matter, and there's ways that in avocados and whatnot, they've done various types of treatments to increase microbial antagonism. But in wildlands, we don't have that option in most cases. So we're really left with exclusion to the degree possible and trying to keep from moving it around further.
I just had one more thing to add, just in terms of, it's not wildland related, but in the nurseries, I know there are antagonistic fungi that have been used to suppress different fungal pathogens, and I know researchers at USDA Fort Detrick have been working with trichoderma, and also at Norris Duck University they've been doing some work, and it's shown good activity against Phytophthora morum. And the last I heard, tentaculata looks like another species that it's active at killing. So not so useful probably in wildland settings, but maybe in nurseries settings as well. So, but it just shows that there are fungal antagonists out there.
Question over here.
Okay,
Yeah, thanks.
wait. Mia had a question which I think is worth asking again.
I was just, because I was thinking where all of our infections are, none of them are in serpentine, and so I was wondering if we were seeing a lot of infections in serpentine, and their ceanothus is in serpentine, and I was wondering if Ted's seen others, just because they are, full of all those toxic nutrients. I thought maybe that would be good to kill Phytophthora.
Well, the best example is ion manzanita is not serpentine. It's ultra acidic, actually. They have pHs down to 3, and Phytophthora is happy there. And what we actually see is general, I think, across our experience, is that poorer soils with low organic matter, whether they're serpentine or whether they're just low nutrient, droughty soils, often those are where we pick up the impacts first. You can see them more readily. And a lot of that probably does is go back to microbial antagonism, which is not operating in a lot of those sites.
Sorry, go ahead.
No problem. Just a simple question about contract specifications. I saw, I might have been in Diana's presentation, doing BMPs and then incorporating basically BMPs into contract specs. Has anybody uploaded an example of that onto one of these great websites where all this information is?
Yes. There are contract specifications at the Cal Phyto website.
Thank you.
In the Golden Gate National Parks, both in Marin County and San Mateo County, we've seen pretty extensive Frangula dieback, and we have gotten positive hits for Phytophthora, a couple Phytophthora species. But, there's also been the drought, and I'm just wondering if that's been seen more broadly regionally, and if this group has any thoughts about whether that might be a Phytophthora-related syndrome.
Where you're seeing this Frangula, is it distributed across wide areas or multiple areas, or?
Yep, across, whole valleys kind of a thing, large landscapes.
Yeah, I mean, one thing, that's the problem with Phytophthora in general, is because they cause water stress symptoms. If we have water stress associated with the drought and we're having mortality associated with that, it looks a lot like water stress and Phytophthora associated with drought or whatever else. And so I think what you have to look at is in sites like that, you've got the same overall symptomology. It doesn't necessarily mean you've got the same thing going on in the whole set. So sites like that, you really have to do enough sampling to confirm that you've got coincidence that's pretty high between your Phytophthora detections and the symptoms. Just in general, and we've been doing a lot of wildland sampling, and Frangula does look bad in some spots, but really, a lot of places where we see Frangula going out, it's because there's Phytophthora there. But a lot of those are riparian situations. If you've got hillside situations, that's a little bit different, but certainly, we have Cinnamomum, Cambivora, there's lots of widespread infestations that go over multiple acres in the Bay Area, Peninsula, elsewhere. So it's not unheard of, and you could certainly have an old established infestation. If you get one big infestation, your potential of having satellite infestations delivered by means of inoculum moving from that site to another one really increase. And that's one reason why we don't want to get big infestations, because they don't just stay in place. They get moved around, and now you have multiple ones. And so that could be a type of thing that's going on there, especially if you have trails and roads and whatnot to connect any of those sites.
Okay, so Allison's asking about coffeeberry dieback. I was wondering, in here, do any of you guys see it? What she's talking about? Someone? Yeah?
Had Steve Chesfold come out and look at the infestation, and since it's all clear, it didn't kill the plants completely. There was entire hillsides, several acres of just the coffeeberry, and it was. I'm not going to remember what it was. It was not Phytophthora, and they've since fully recovered. But I was totally freaked out.
So Steve's here. Steve, do you have anything else to?
Various finds on Frangula. So Laura's the one to talk to from UC Berkeley, from Matteo Garbalato's lab.
You might notice that we just go from one pathologist to another.
So there is going to be further study of Frangula and other susceptible species in wildlands starting next month, I believe that Steve is referring to. So that's upcoming. If you have burning questions, you may write them down. I'm going to put this outside on a table over there in the other room, and we'll be gathering them, collecting them for the discussion after lunch. So now there's a break, and there's the pathogen smoker, and Janelle is going to gather the group. I'm going to let